Module 1, Objective 14, explain the primary, secondary, tertiary, and quaternary structures of a protein. Include an explanation behind the forces that hold each structure together. Wrong direction. In terms of protein shape, the primary structure is nothing more than the sequence of amino acids within the polypeptide. So as we can see here, which amino acid is first, which is second, which is third, fourth, so on and so forth. So it is nothing more than that sequence. And remember, there are 20 different amino acids. So it's almost like we use 26 letters in order to create this word primary. The P comes first, followed by the R, followed by the I, followed by the M, followed by the A, followed by the R, followed by the Y. In the same fashion, a protein and the primary structure of a protein is nothing more than which amino acid is first, which is second, which is third, so on and so forth. What dictates that pattern or that recipe? Well, it's dictated by the DNA. The DNA holds the recipe for the proteins within our body. So within the code of the DNA, it actually reads which amino acid is first, which is second. And just like any recipe, the ribosome follows the instructions, putting the amino acids in a particular order, and in the end, you get the polypeptide and then protein. So the primary structure is nothing more than the sequence of amino acids. What holds the primary structure together? These peptide bonds. So in between each of these amino acids, we have the peptide bond. And remember, the peptide bond is just the name given to a particular type of covalent bond found between amino acids. Proteins are often not linear, but they tend to be globular. In fact, if you were to look at a three-dimensional protein, and I'll jump ahead to an image, you can see here they're quite complex, and they have a lot going on. So in order to build the functional protein, we have to go through these individual steps. So again, primary structure, nothing more than the sequence of amino acids dictated by the DNA. The secondary structure is when you start getting some shape, and there's two main shapes within the secondary structures. We have spirals and pleats, and this image represents a spiral, and you can see how that spiral occurs. It's almost like the old phone cords, if you will. A pleat is more angular like this. So these are the two basic structures found within the secondary structure of a protein. These are held together via hydrogen bonds. So while peptide bonds hold primary structure, the hydrogen bonds form the secondary structures. The tertiary structure is when you get further folding and you get more of a three-dimensional shape. And tertiary structures are due to the chemistry of the R group. So let me just sort of dictate this. What holds the tertiary structure together is the chemistry of the R groups. Remember that each amino acid has that variable group, and we call it the R group. So here we have the R groups in this chain, and it's just the chemistry of those. So let me give you just one or two examples of such chemistries, and you can go from there. If this were a positive, let's say this particular R group had a positive charge and this had a positive charge, what do we know about two positive charges? Well, they repel, and in repelling you may cause the molecule to take on a shape such as that, where they're sort of forced apart. And that's going to further produce this three-dimensional shape that we sort of identify as the tertiary structure. Another example, if this particular R group was hydrophobic, what does hydrophobic refer to? Fear of water. Um, this particular R group might tuck itself deep inside of this molecule, causing again some type of conformational change in shape, giving rise to a further enhanced or more detailed three-dimensional shape. So this is the tertiary structures. All proteins have primary, secondary, and tertiary structures. Some proteins have a quaternary structure. And quaternary structures occur when you have a protein that has multiple subunits. This particular protein is hemoglobin. It's found within our red blood cells, and it carries oxygen. It actually has 4 subunits, 1, 2, 3, 4. So these are 4 proteins that are created, and then they come together in order to form the quaternary structure of hemoglobin. So quaternary structure is a final protein complex produced by the interacting polypeptide chains or subunits. And again, not all proteins have a quaternary structure, only those that are made up of multiple subunits. Extreme temperature and pH can actually cause a denaturation of a protein. Here we have a protein with all of its complex shapes that are held together by various types of bonds, and extreme temperature or pH can cause a denaturation where you lose those the bonds between those various levels of protein structures, and your protein falls apart. This is what happens at about 105 degrees Fahrenheit in our bodies. When our temperature rises to 105, our proteins denature. They fall apart. If that protein was an enzyme, it loses its function. And of course, since enzymes control metabolism, without metabolism you would quickly die. So 105 is a very serious temperature for an adult. Proteins, if not destroyed through denaturation, can renature through a process called renaturation. And they can actually regain their form once the temperature or the pH has been corrected. So that is denaturation and renaturation.